Hello, my name is Jaden Bender and this is our Gut Microbiome Project. For introduction, the gut microbiome in general is the different types of bacteria in the body's gut. It is closely related to the health in the body. One physiological condition that is related to the gut microbiome is type 2 diabetes. These two are related because type 2 diabetes is a disease that causes an imbalance in blood glucose levels. The gut microbiome may reshape intestinal barrier functions and host metabolic and signaling pathways. This can directly or indirectly cause the rapid progression of insulin resistance in the body. There are thousands of different microbes in the gut, and with certain dietary factors, it can strongly affect the gut microbiome composition and promote diasobosis. Their relationship is correlative. Another physiological condition that is related to the gut microbiome is autism. These two are related because of the brain-gut link. A study was ran on rats that did not show signs of autism, and when they were introduced to feces from humans with autism, they responded by showing signs of autism. While this does not prove that the bacteria in the gut can cause autism, it does show that makeup of the gut can contribute to hallmark features of this disorder. The way this changes behavior is, since the brain and gut are connected, it changes the way the brain makes different proteins. Their relationship is also correlative. Our first question asked if health had an effect on the gut microbiome of dogs. Our hypothesis is if the dog is healthier, then there will be more bacteria present in the gut microbiome. And our second question asked if age would have an effect on the gut microbiome of dogs. And our hypothesis is if the dog is younger, then it will have more bacteria in the gut microbiome. For materials and methods, round one, we sampled 751, which was a 12-year-old, 11-month-old female border collie blue heeler mix of poor health, and it was obese with cancer. Our second sample was 683, a 9-year-old male beagle of poor health, and it was obese. Our third sample was sample 458, a 6-year-old male shepherd hound mix of very good health and no health issues. And our last sample was a 12-year-old female West Highland Terrier of very good health. And our protocol was following the Bio 200 lab manual. First, we used purification of DNA from fecal matter using the QIAGEN kit according to manufacturer instructions. Number 2, quantification of DNA in samples. And number 3, qPCR setup of amplification of DNA. Next was purification of DNA from fecal matter using the QIAGEN kit. We weighed out about 0.2 grams of the sample into 2 milliliter centrifuge tube and placed it on ice to prevent degradation of the DNA. Then we added 1 milliliter of Inhibit X buffer to each sample and vortexed it until it was thoroughly homogenized. Inhibit X removes inhibitory substances by separating them from stool samples. Next, we used heat suspension for 5 minutes at 70 degrees Celsius, vortexed it for 15 seconds after 2 minutes, then continued heating up for the remaining 3 minutes. Heat lyses bacteria cells in stool at 70 degrees Celsius. Then we centrifuged to pellet stool particles, pipetted 500 microliters of the supernatant in a centrifuge tube with 1.5 milliliter proteinase K enzyme that cleaves peptide bonds. Then we added 500 microliter buffer AL reaction buffer to proteinase K, incubated it at 70 degrees Celsius for 10 minutes. Then we added 500 microliters of 96 to 100% ethanol to lysat and vortex to allow binding of DNA to spin column. Then we transferred 750 microliters of the lysat to spin column of centrifuge for 1 minute at 14,000 RPMs. Then we discarded the contaminating solutes in liquid that flowed through. Next, we opened the spin column and washed 500 microliters of AW2, which removes impurities, centrifuge for 3 minutes at 14,000 RPMs twice to discard filtrate, and we discarded filtrate each time. Then we transferred spin column into new microcentrifuge tube and added 200 microliters of buffer ATE onto spin column membrane. That disrupts DNA interaction with membrane and allows DNA to flow through with buffer. And then we incubated it for 1 minute at room temp, centrifuged it for 1 minute to elute DNA. Next was quantitation of DNA via nanodrop spectrophotometer. We utilized nanodrop 2000 software nucleic acid DNA. DNA absorbs light at 260 nanometers and proteins absorbs light at 280 nanometers. We use this information to calculate concentration of DNA and determine ratio of absorbance at 260 nanometers to 280 nanometers, a measurement of purity. Then we loaded 1 microliter of diluted water onto lower pedestal, lower arm, and performed routine verification. We wiped upper and lower pedestal with Kim wipe between each measurement. Then we changed micropipette tips between each sample. Then we loaded 1 microliter DNA elution buffer onto lower pedestal, lower R, and measured blank. And for each sample, we loaded 1 microliter of the sample onto the lower pedestal, lower arm, and measure concentration of 260 to 280 nanometer ratio. For results, for sample 683, we got an absorbance of 0.95, a ratio of 1.43, and a concentration of 47.5. For sample 458-H, we got an absorbance of 0.042, a ratio of 2.0, and a concentration of 2.1. For sample 571, we get an absorbance of 0.082, a ratio of 2.69, and a concentration of 4.1. And for sample 318-H, we got an absorbance of 0.150, a ratio of 1.66, and a concentration of 5.2. Our CQ values and relative abundance of bacteria versus fungi. Our positive control B-CQ was 13.43. Our B antilog was 9.08 times 10 to the negative fifth. Our F-CQ was 31.71. Our F-antilog was 2.84 times 10 to the negative 10, and our ratio was 0.42. Our negative control was our B-CQ was 27.67. Our B antilog was 4.68 times 10 to the negative 9, and our F-CQ was 34.08. Our F-analog was 5.52 times 10 to the negative 11, and our ratio was 0.81. For sample 683, our bacteria was 22.00. Our bacteria analog was 2.38 times 10 to the negative 7. Our fungi CQ was 33.55. Our fungi analog was 7.97 times 10 to the negative 11, and our ratio was 0.66. For sample 458, our bacteria CQ was 26.33. Our bacteria analog was 1.18 times 10 to the negative 8. Our fungi CQ was 35.42. Our fungi analog was 2.17 times 10 to the negative 11, and our ratio was 0.74. For sample 5.71, our bacteria CQ was 25.28. Our bacteria analog was 2.46 times 10 to the negative 8, and we did not get any fungi values. For sample 318, our bacteria CQ was 26.43. Our bacteria analog was 1.1 times 10 to the negative 8, and our fungi CQ was 32.35, and our fungi analog was 1.83 times 10 to the negative 10, and our ratio was 0.82. For round 2 and 3, our animals compared were sample 1057, a 4-year-old, 2-month-old female pit bull of very good health. Sample 591, a 5-year-old, 3-month-old male pug of average health. Sample 3.21, a 10-year-old, 6-month-old female boxer of average health. And sample 340, 8 years, 10 months, female boxer with very good health. Our samples were chosen from other groups due to valid CQ values, which were under 25. Samples chosen from other groups due to valid CQ values, which were under 25. Round 1 samples produced CQ values over 25, indicated absence of bacterial DNA, may have indicated contamination of samples or error during DNA extraction, did not perform second DNA purification or quantification via nanodrop, and only performed additional qPCR measurements of phylum of bacteria. Our new question: does the age of dogs affect their gut microbiome? Phyloprimers for round 2 were Acinetobacteria and Firmicutes, and for round 3 it was Actinobacteria and Bacteroidetes. Our CQ values and relative abundance of round 2 were, for CQ values we had, for 1057 we had 32.38, 1.23 times 10 to the negative 10. We had 24.46 and 4.339 times 10 to the negative 8. And our ratio was 2.83 times 10 to the negative 3. For 591, we had 31.43, 3.307 times 10 to the negative 10, 23.37, 9.222 times 10 to the negative 8, and 3.586 times 10 to the negative 3. For sample 321, we got 3.77, 5.478 times 10 to the negative 10, 24.03, 5.855 times 10 to the negative 8, and 9.356 times 10 to the negative 3. For sample 340, we got 29.73, 1.12 times 10 to the negative 9, 25.02, 2.933 times 10 to the negative 8, and 3.819 times 10 to the negative 2. And our CQ values for relative abundance of bacteria to 2 bacteria file around 3. We got 1057 for 27.13, 6.81 times 10 to the negative 9, 30.54, 6.39 times 10 to the negative 8, and a ratio of 1.10657. For sample 591, we got 29.22, 1.60 times 10 to the negative 9, 26.49, 1.06 times 10 to the negative 8, and 0.15094. For sample 321, we had 29.17, 1.66 times 10 to the negative 9, 3.09, or 30.09, excuse me, 8.73 times 10 to the negative 10, and 0.52590. And finally, for sample 340, we got 29.16, 1.66 times 10 to the negative 9, 26.47, 1.08 times 10 to the negative 8, and 0.65060. And for our discussion, extracting the DNA involves measuring the absorbance at 260 nanometers ratio and concentration. A concentration of greater than 1 identifies higher enough concentrations to be able to dilute the solutions. All concentrations recorded were greater than 1. The ratio helps identify purity among DNA, absence of RNA. Samples 458 and 318 in the first round had ratios between 1.5 and 2, therefore a greater purity among the 4 samples and less RNA present. The negative control in the first round had a BCQ of 27.67 and FCQ of 34.08. These numbers showed no DNA to be found, which is accurate due to the no addition of extracted DNA or controlled DNA. The positive control in the first round of qPCR had a BCQ of 13.43 and an FCQ of 31.71. Both of these numbers should have been under 25 due to the addition of controlled DNA knowing the exact value that should be present. We did not conduct a positive control for round 2 or 3. And our discussion continued. Negative control in round 2 and 3 of the qPCR were values well over 25, as stated before, that is expected due to no addition of DNA. Differences among the gut microbiome did not vary as much as we would have liked. Our 2 younger dogs had bacteria present in their microbiome like we expected, but so did one of the older dogs. The younger dogs had CQ values of 24.46 and 23.37, whereas the older dogs had values of 24.03 and 25.02. With that being said, our results did not support our hypothesis that if a dog is younger, then it will have more bacteria present in our gut microbiome. And our references are here. Thank you.